Good morning, students. Welcome to online biology lesson 5. Today, our topic is transport of material through the plasma membrane. Before that, mm, just have a quick view of the structure of plasma membrane. So here on the screen, you are seeing the structure of the plasma membrane. In our earlier class, we already studied of the structure of plasma membrane. But let's have a quick review. Now you can go by the page number 13 in your material. The page number 13, you are seeing the structure of the plasma membrane. So on the screen, you are seeing the structure of the plasma membrane. So this model is a current accepted model, which is called as fluid mosaic model. This model was proposed by Singer and Nicholson in 1972. So according to their model, so this membrane, plasma membrane, was composed of lipid bilayer, means there is two layers of lipids. This is one layer and this is another layer. This is lipid bilayer, means two layers and containing mobile proteins. So here you are seeing these are mobile proteins. So these are mobile proteins. These mobile proteins help in the transport of materials, ions, and atoms across the membrane, cell membrane. Okay, this is the basic structure of uh, plasma membrane or cell membrane. So why we did have a look for this time, because depends on the structure. Our next topic is gonna be continue depends on the structure. So that's why we had a quick uh, view on the structure of plasma membrane. Okay, then let's go for our topic. Today's topic is transport of materials through the plasma membrane. You can go for the page number 33 in your material, the page number 33. So, transport of materials through the plasma membrane. This cell membrane or plasma membrane, also known as selectively permeable membrane. In our previous lessons, you already learned this word, selectively permeable membrane or semi-permeable membrane, because so this cell membrane allows only some substances inside of the cell. Okay, that's why so this cell membrane or plasma membrane known as selectively permeable membrane or semi-permeable membrane. So by having of this nature, this uh, plasma membrane controlling what substances may enter or leave the cell, means it will control which substances can enter or which substances can leave from the cell. So materials are conducted in and out of the cell by two methods. So these are the materials, ions, atoms, sometimes water molecules are moved in and out of a cell by two basic methods, means the movement of these materials in and out of cell by these two methods. There are the two basic methods. which explains the movement of these materials in and out of a cell. Those methods are, one is passive mechanism and the second one is active mechanism. This passive mechanism, they don't need any energy. Without using any energy, the movement of materials occurs in and out of a cell. When it comes about this active mechanism, the movement of materials in and out of cell by using energy. What is the energy here? That is the ATP. Means in briefly, active mechanism uses energy. Passive mechanism they don't need any energy. So these two mechanisms explains or give the information about the movement of materials in and out of the cell. Well, let's go for the passive mechanism. The first mechanism is a passive mechanism. So you must remember this passive mechanism do not need any energy. Okay, this is passive mechanism. So here, this passive mechanism is the movement of molecules from an area of high concentration to an area of low concentration without use of energy by cell. So as I told you earlier, so this mechanism explains the movement of molecules from high concentration to an area of low concentration without use of energy. Okay, well, these three passive mechanisms have three forms. So these three forms are, one is diffusion, second one is facilitated diffusion, third one is osmosis. Diffusion, facilitated diffusion, osmosis. These are the three forms of passive transport. Well, let's have a look of diffusion. So, diffusion is, or we can call this diffusion as a simple diffusion. So, diffusion is the movement of ions, atoms, or molecules from regions of high concentration to regions of low concentration. Here in this picture, on the right hand side, you are seeing a picture. So here in this picture, this part have high concentration of molecules and this part has low concentration of molecules. And this the line, what you are seeing in the pink color, just assume as a like a barrier line. So here the movement of the molecules from high concentration to low concentration is we call as diffusion. Okay, we call as a diffusion. Sorry, here there is a small mistake. That is a diffusion. So diffusion is the movement of ions, atoms, or molecules from regions of high concentration to regions of low concentration. Here, so once the diffusion occurs, so you are seeing here the equal concentration in the both sides. So this diffusion occurs in both living and non-living cells. So let me give an example for this diffusion. Okay. So when you take a perfume bottle, inside of this perfume bottle, it has the concentration of perfume. So when you open this perfume bottle cap, when you spray into the air, these high concentration perfume molecules spread all over the room because so here this perfume molecules, high concentration perfume molecules will move to the low concentration of low concentration area. In the same way, you can see, you can do in your home this kind of experiment. Take a tea bag, okay, and put it this tea bag in a cup of hot water. Then slowly you can observe, like slowly the color of the tea slowly will spread into the water. So means the tea bag it have the high concentration of the tea, and slowly when you drop this tea bag into the water, this tea slowly move to the low concentration area like water. So even though you can do this experiment at home, take a drop of ink when added to a glass of water. Just take a glass of water, put a drop of ink, and slowly you can observe this ink spread slowly throughout this glass of water. This process we call as a diffusion. Diffusion is nothing but you have to remember the movement of ions, atoms, or molecules from region of high concentration to the region of low concentration. So this we call as a diffusion. So some molecules, some atoms will enter into the cell by the process of the diffusion. This cell membrane allows these molecules because of the this diffusion. It's a kind of force. Okay, the next one is second type of the passive mechanism is facilitated diffusion. So in this facilitated diffusion, the carrier molecules. What are these carrier molecules? In a previous picture, like we already saw the picture of this structure of plasma membrane. In this structure of plasma membrane, you already saw, so these are the carrier proteins, the proteins. So these carrier proteins in the cell membrane accelerate the movement of relatively large molecules from high to lower area of concentration. For example, let's say there is a high concentration of some ions are here, okay, or large molecules. And when comes about here, there is a low concentration of the same large molecule. So at the same time, what happens? So these carrier molecules means these protein molecules catch these large molecules and help in the movement of these large molecules from high concentration to low concentration. So the same way, the carrier molecule in the cell membrane accelerate movement of relatively large molecules from high to lower area of concentration. So here, glucose molecules are the best example. So they move across the membrane by the process is facilitated diffusion. So these glucose molecules are large molecules. So these large molecules enter inside of the cell by the process we call as facilitated diffusion. Not only this glucose molecule, some other large molecules also enter inside of the cell through the a process we call as facilitated diffusion. So this is the second passive mechanism method. The first one is simple diffusion and the second one is facilitated diffusion. So in a simple diffusion only depends on the concentration, the ions or atoms or molecules enter inside of the cell across the cell membrane. Here in the facilitated diffusion, they use these carrier molecules. So these atoms or ions or the big size molecules, they use the carrier molecules. What are the carrier molecules here? So here you are seeing on the screen, these carrier proteins or carrier molecules. So you can see here, it catches one large molecule, okay? So then slowly it will release, step by step, it release these molecules inside of the cell. So in a facilitated diffusion, they use carrier molecules. So these carrier molecules are nothing but protein carriers or protein molecules which are present in the cell membrane. Okay, next let's go for the third passive mechanism method, that is osmosis. Osmosis. Go for the page number 34, then you can see the osmosis. So this process is the diffusion of water across the simple semi-permeable membrane. Means here, this osmosis explains the movement of water molecules across the semi-permeable membrane from the area of low concentration to the area of high concentration. So this osmosis occurs until the equilibrium takes place between the solutions. So here you are seeing the words hypotonic solution, hypertonic solution, and isotonic solution. So here, let me give an example. So normally, uh, if you mix, let's take 2 cups of sugar, sorry, 2 cups of water. So let's say cup A and other cup is B. So you can add 3 spoons of sugar to the cup A, and you can add 1 spoon of sugar in cup B. When you compare these 2 cups of water, which cup is which water is more sweetened? Means cup A, because here the concentration of the sugar is high when you compare with the other cup water, like cup B. So means here, because of this high concentration solution, so this we call as hypertonic. Hypertonic. And here when come with the cup B, so it having the low concentration of the sugar, means the less sweet. So this we call as hypotonic. Hypotonic. So this way we can define what is hypertonic solution and what is hypotonic solution. Hypertonic solution, hypotonic solution. So a solution which has a high concentration, this we call as a hypertonic solution. A solution with the low concentration we call as hypotonic solution. So here osmosis takes place. It's quite opposite to the diffusion. So in diffusion, we learned that the movement of molecules from high concentration to low concentration. So to remember this diffusion, just put it in your mind, perfume bottle. This perfume bottle have the high concentration of the molecules. When you spray this perfume, so this high concentration of the molecules will spread towards the lower concentration of the area, lower concentration area. But here, when comes about the osmosis, osmosis is quite opposite to the this diffusion. So in osmosis, you can see the movement of water molecules. Only we are saying the word here, the movement of water molecules across the semi-permeable membrane from the area of low concentration to the area of high concentration. It's opposite to the diffusion, but you must remember we have to use a word across the semi-permeable membrane. So this word gives the exact meaning of this osmosis. So the movement of water molecules uh, across the semi-permeable membrane from the area of low concentration to the area of high concentration. So this osmosis occurs until the equilibrium takes place between the solution. Means, as I told you, solution A is a hypotonic solution and solution B is a hypotonic solution. So the movement of water molecules from low to high will occur this through the semi-permeable membrane. So this will occur until this will become as a like uh, low concentration solution, like hypotonic solution, means until equilibrium takes place. Okay, so here we're gonna learn some words like osmosis can result in two extreme conditions. So one is plasmolysis and second one is deplasmolysis. So here, when you put a plant cell in an isotonic solution, there is a no change in their cytoplasm. The cell remains still like a normal. When you put a plant cell in hypotonic solution, from outside the water will enter inside of the cell because here the concentration of this cytoplasm is higher than this solution. So due to this reason, the water, because we are still talking about the osmosis, due to the osmosis, so the water try to enter into the cell. So slowly the water will enter into the cell and the cell will get swollen, but it will get swell and slowly the water is entering and finally the cell will like explode or the cell wall will get damaged. Okay, this process we call as deplasmolysis. Deplasmolysis occurs when a cell placed in hypotonic solution, means when you drop a cell in the hypotonic solution, so the water will enter inside of the cell and so due to the excess of water get inside of the cell will get swell and finally the cell will explode or the cell will die because of the this breakage of the cell wall and the cell membrane. So this process we call as deplasmolysis. And the second one is plasmolysis. So when you drop a cell in a hypertonic solution, so here this solution is having the high concentration of the solution, like high concentration. So when you drop a cell into the high concentration solution, so according to the osmosis, the water from the cell will go out and slowly, so the cytoplasm gets shrink like this. It will get shrink. So this we call as a plasmolysis. I hope you understand this, what is plasmolysis and deplasmolysis. Let me give a quick idea about this both. So osmosis can result in two extreme conditions, like when you drop a cell in a different solution. So when you drop a cell in the isotonic solution, means there is a normal solution, the cell will remains as a normal cell because both of this cytoplasm and the solution concentration is equal. So that's why the water will enter and go out and the cell remains normally. When you put a cell in the hypotonic solution, means the solution concentration is lower than the concentration of cytoplasm. When you put a cell in the hypotonic solution, so the water, according to this osmosis, the water from low concentration to high concentration, okay, water molecules will move from here to here, and because of this excess water, so the cell will get swollen, and if keep on this water will getting inside, so finally the cell will explode or it will cause the death of the cell. Then the third one is, sorry, this process we call as deplasmolysis. And this one is when you drop a cell in a hypotonic solution, so the solution concentration is higher than the cytoplasm concentration. So according to the osmosis, the water in the cytoplasm will go out from the cell to the high concentration solution. Due to this reason, the cytoplasm will get shrink. So this we call as plasmolysis. Okay, then this plasmolysis and deplasmolysis result from the combination of two important forces. So this process causes the two important forces. One is osmotic pressure and the second one is Turger pressure. Okay, what is osmotic pressure? A solution with a high solute concentration has low water concentration. Therefore, it has high osmotic pressure because water always tends to move towards solution of greater osmotic pressure. Okay, let me give an example. This is a cell, and let's say this is a small vessel or just a cup. So here, let's say this solution has high concentration and the cytoplasm of the cell has low concentration. So here, now you can imagine, according to the osmosis, the water molecules will move from low concentration to high concentration across the semi-permeable membrane, isn't it? So in this situation, so when you drop this a cell in high concentration solution, so now according to the osmosis, the water from the cell will move to this high concentration solution, isn't it? So this kind of the situation we call as osmotic pressure, means now the cell has a high osmotic pressure because here water always tends to move towards solution of greater osmotic pressure. Like, so here the water always moves, try to uh, towards the high concentration solution. So here, this kind of the force or pressure we called as osmotic pressure, means the cell has the high osmotic pressure, means the water tends to move towards the high concentration solution. So I hope you understand. Let me give one just brief example again. So let's say this is a cup which have the solution, high concentration solution. Okay, high concentration solution. And when you drop a cell, let's say this cell cytoplasm has low concentration of like low concentration cytoplasm. When you drop a cell here, so the water molecules automatically move from low concentration to high concentration across the semi-permeable membrane. So force, this force which can tend to move towards the solution of greater osmotic pressure, this pressure we call as the osmotic pressure. In this case, so this cell has high osmotic pressure because the water will move from low concentration to high concentration. This we call as osmotic pressure. And the second one is turgor pressure. This turgor pressure is excreted by water against the cell membrane or cell wall of the plant cell. Okay, let me give an example. So this is a beaker or a cup. So in this cup, the solution is low concentration. So when I drop a cell, this cytoplasm of the cell has high concentration. Now what will happen? The water molecules will move from low concentration to high concentration, isn't it? So when the water is moving into the cell, so this can cause the this cell walls shows the this mm, pressure like a force to avoid the breakup of the cell. So this pressure we call as the Turgor pressure. So this is the example hemolysis to the erythrocyte when it's placed into the dilute solution. Okay, students. So this is the class for today. I hope you understand. So if you have any questions, try to ask these questions to my WhatsApp. To WhatsApp, I think you guys have my WhatsApp number, or you can ask that directly. They will give my WhatsApp number. You can ask any doubts from there. Thank you very much. Have a good day. Have a safe stay. Thank you.